Good morning. I'm Ramachandriya from a PhD student from Biosystems and Agriculture in the department. And today I'm going to discuss my PhD research, a part of it, which is on pretreatment of Eastern Red Cedar for bioethanol production. So we're starting with biofuels. Eastern Red Cedar is called as invasive, or it's a nuisance tree because it's kind of widespread all throughout half of the United States and also it's into the United States. We have a problem with this tree. This tree is invasive. It likes to infest grasslands, especially like in the Central Plains, and therefore the governments have to, the local governments have taken steps to get rid of it, because due to the presence of Eastern Red Cedar, there's loss of pasture land, there's loss of water from the ground table, and also for people like me, there's a lot of the pollen that gets released from the tree causes a lot of allergy. The solution is to convert the biomass into something usable so that it benefits the economy. I know Dr. Hizroglu is working on development of particle boards, fence posts, and other materials. Dr. Renford works also with cedar oil for perfumes and other specialty work. But this project is going to look on conversion of the polysaccharides that is present in Eastern Red Cedar into ethanol. By ethanol, we all know that we have a problem with the transportation sector. Our over-dependence on oil has resulted in research into development of alternative fuels such as ethanol. So this research is a step to see how we can develop a process for the production or the biorefinery of Eastern Red Cedar into ethanol. You see the ethanol production potential from different softwares. I forgot to mention that Eastern Red Cedar is a software. What that means to us is that Eastern Red Cedar is going to be really challenging, because by nature, softwoods have high amount of lignin, and also the nature of lignin, which is a polymer in the wood, that creates a lot of difficulty in the bioconversion process. If you compare the potential of Eastern Red Cedar with other softwoods, such as Douglas fir or spruce, you can see that from 1 ton of biomass, we produce almost 395 liters of ethanol, whereas we compare it with Douglas fir, it's about 5% less, in comparison with spruce, about 10% less. If you want to compare this with switchgrass or other energy crops, they also produce about 435 liters of ethanol theoretically. So it's quite comparable. The yields are quite comparable with the energy crops and other softwares. Biomass pretreatment. This is the first step during the bioconversion process. This is a step where we take the biomass and we convert it in a way so that the biomass is amenable for the subsequent processes. We are looking into acid bisulfite pretreatment. This is a pretreatment process that has been patented by the University of Wisconsin and U.S. Forest Services. This is a modified version of biosulfite pulping that used to be there long back, and it's also there nowadays. The two important chemicals that are used in the process are sulfuric acid and sodium bisulfite. During the process, what happens is that these chemicals react with the biomass components and result in delignification and removal of hemicellulose, which is again a component of biomass, that results in making the biomass more amenable for the enzymes to come and work in the subsequent process. And this is how the whole this is what happens during the pretreatment process. We have cellulose fibrils, which is in this blue color, and there are hemicellulose green color overlaid with, and interacting with both lignin and lignin, which is there outside. And what happens in the pretreatment process is basically breaking that lignin and chopping that hemicellulose off, so that the enzymes can now go through and access cellulose fibrils and produce glucose that could further ferment into ethanol. So the overall objective of this project is to develop a process to convert the polysaccharides, particularly the cellulose and if possible hemicellulose, into ethanol. But the first step in this research is to optimize the pretreatment step, because because overall the success of the overall bioconversion process depends on the first step, because it subsequently affects the the following steps. During our preliminary research that we conducted at our labs, we found out that at 200 degrees Celsius and with the chemical loading that we usually use, there was a lot of loss of glucose. And gluten, which is coming from cellulose, is the substrate for ethanol. So if we have loss in gluten, that means that we are having a loss in the overall yields of the process. So the objective of this research was to come up with a process that would prevent the loss of gluten. From literature, we know that cellulose hydrolysis, because we have acids in it, and at high temperatures, as 200 degrees Celsius, and if the time is really high, then it basically affects. It starts breaking down the cellulose. The acids break cellulose into glucose. So that's how we see the glucose loss. Moving to the materials and methods. This is where we conduct the pretreatments. This is called a pressure reactor. We put biomass into the vessel, and we load it with a pretreatment liquor, which has sulfuric acid and sodium bisulfite at these concentrations. Then we have the outer jacket that heats it up. We have an impregnation for 3 hours at 90 degrees Celsius. At the end of impregnation, we ramp the temperature up to 200 degrees Celsius, hold it for a particular time, and in this case, we're trying to vary the time. And then at the end of that whole time, we cool the reactor and separate the solids and the liquids. And then the next step we take the biomass into these reactors and try to test whether our pretreatment process works or not using enzymes. For that, we use accelerated 1500 enzyme produced by General Board at a loading of 0.5 ml per gram of gluten. And these reactors are generally buffered at 0.05 molar of sodium citrate, and those are the pH temperatures and RPMs that we use. We do compositional analysis using National Renewable Energy Laboratory protocols. The composition for composition analysis, we use HPLC, which where we have a carbohydrate. We have a column which analyzes carbohydrates. And for this particular experiment, I use statistics to draw conclusions. Use SAS 9.2 drug GLM procedure, and for the post-hoc analysis, I used LSD. Moving on to results, this result shows, this graph basically shows gluten to glucose conversion at different hold times. What this basically means is that if this was 90%, the second one, which is a 10 index, that means that we could convert 90% of the gluten that was there in the material into glucose. That's a very good sign. So at 10 minutes and above, we get good conversions over 80%. But at 5 minutes, it's lower. That's because we've not at 5 minutes we've not had sufficient time for the reaction to occur. That's why the conversions are low. Now, when I'm talking about gluten conversion in this particular graph, I refer to gluten in preheated biomass. I'm not referring to gluten which is there in the original biomass. Now, let's move on. Gluten loss. Gluten loss is during a pretreatment process. So if we see the composition of the raw substrate and compare it with the preheated substrate, then to do the mass balance from the HPLC, we can basically find how much of sugar is left with the substrate. So this is a graph which shows the gluten loss of a preheating process. As you see, as we increase the temperature, there's an increase in gluten loss, which makes sense because at that high temperature and if we provide sufficient reaction time, then the acid present during the reaction is basically reacting with glucose or cellulose and breaking it down. So we're losing glucose during these pretreatments. Okay. So at 5 is kind of negative, but that's because of our of some error in determination. Let's move on. This is the most important result. Although we saw in previous slides that during an enzymatic hydrolysis step, the conversions were high. But what this is showing is basically wood to sugar ratio. This means that if it was 85%, that means 85% of glucose that was present in biomass could be converted into glucose. So 85% of the glucose is basically available for fermentations, which is good. As we see, at 5 minutes we have around close to 60% yield. The yields get higher at 10 minutes. So that's the best time. And above it, the yields decrease because there was loss of glucose. There was a lot of loss of glucose, as we saw from the previous slide. So in conclusion, under these testing conditions, the digestibility of the material was above 80% for all conditions, above 10 minutes. And by digestibility, I mean the efficiency of enzymatic control system. More than 40% glucon loss was observed for treatments with hold time of 20 minutes and more, so which is not good. So 10 minutes would be the best condition. I also had lignin removal data, but I didn't show you. But generally, the amount of lignin that was removed from the biomass was ranged between 30 to 76%, which is very good because from this process now we can produce lignosulfonates, which has a market value. Highest wood to sugar yield was 85% attained at 10 minutes, and at that period of time, we just had a glucose loss of 6% and a lignin loss of around 61%. So our research hypothesis was right by decreasing hold times. We could prevent loss of sugars and thereby result in an increase of wood to sugar ratio. With that, I'd like to acknowledge my advisor, Dr. Perkins, and my committee members, Dr. Dunford, Dr. Isragulu, and Dr. T, lab managers, graduate students, and the funding agency, South Central Sun Grant Initiative. That, I'd like to thank you all, and I'm ready for questions.